first off, I want to start off by showing you our Star Navigator, which is a handy tool to determine what application that is provided by Star that would fit your needs the best. So for instance, when working with proteins, you would want to look at the proteomics section, which we provide 4 different applications to work with proteins. You can use just the simple explanations here to determine what application you might be interested in, or you can also hover over any of them. That'll have a balloon pop up that provides more information about the application, as well as links to videos and FAQs, as well as our tutorials and any data that might be of use to you. But more importantly, you can also launch any of our applications from the Star Navigator. So I'll go ahead and launch Protean 3D from here. So this is what Protean 3D looks like when you initially open it, when you don't have a file open. So you can easily get started with Protean 3D for our integration and structure visualization and sequence analysis by using the Getting Started section of the Details panel, which you can open a file or the motion library, as well as watch a training video or any of your recent documents that you've had open. You can also do any of these same activities using the File menu and do File Open. You can also use Open Group to open multiple files at once. So for instance, if you find a structure or paper that has a structure that you're interested in and you want to be able to visualize it in Protean 3D, you can first go download the structure from the PDB website using any sort of web browser. So RCSB PDB provides a data bank of all structures that are solved in the United States at least. There are also other websites for other countries, and we support those formats as well. So today I want to work with a PDB called 7TIM. I can search for that PDB using the PDB ID, or I could use the name of the molecule or an author or something along those lines in order to help expedite my search. So I'm just going to search for 7TIM because I happen to know the PDB extension number. On that page, I can then download the file that I need using this drop-down menu. And Protean 3D easily opens PDB file text or PDB file GZ, as well as the biological assemblies. For today, I'm just going to simply download the PDB text file and save it to my downloads folder. I can then, once it's been downloaded, I can then easily return to Protean 3D and use the File Open to open the file from the Downloads folder. And simply click Open. Once the file has been opened, this is the general workbench for Protean 3D. Before I get too far into it, I want to explain a little bit about what I will be referring things to as. For instance, the structure, analysis, and sequence are all views, whereas the explore, methods, details, style, and models are all panels. And within the panels, there are expand bars that provide extra information such as the molecules expand bar or the features expand bar. Additionally, this workbench can be easily customized to however you would like. For instance, you can minimize these side panels and then have them pop out for you when you want them to work, or you can maximize them and put them back to where they were. Additionally, by clicking and dragging, you can move any of the panels you would like, and the black box then indicates where they will be going. So I intend to move the details panel over to the left-hand side in order to be able to see the information that is provided there more easily. However, I don't need it quite so wide, so I can easily just use this arrows to make it skinnier and a little bit more out of my way. So the details panel, when you first open a structure, provides useful information that is provided within the file. For instance, the title as well as the classification of the protein and the technique in which the protein structure was solved using. For instance, this one was solved using X-ray diffraction with a resolution of 1.9 angstroms. You can also easily link to the PubMed ID or reference on NCBI's website, or you can link back to the PDB website and see that file again present on their website. So the first thing that I tend like to do when I first open a structure is I like to be able to interact with it and see what it looks like. So you can easily do that using our interactive structure view. By default, we have our structure view set on free rotation. And all you need to do is click and drag, and the structure will move in any direction that you please. All I'm doing is holding down the mouse and clicking and rotating, moving my mouse around. Also, at any time, you can zoom using the scroll function on your mouse by scrolling in and out to zoom. If you prefer to rotate simply on the Z-axis, you can switch the toolbar to this icon, and that will pin the center and only allow you to rotate on the Z-axis. You can also simply move the structure without doing any sort of rotation using the 4-arrow icon, and then that allows you to adjust where your structure will be located. If you prefer not to use the scroll function on your mouse, or you don't have one, you can also use the zoom function with the magnifying glass and just move your mouse up and down to zoom. Also in the toolbar, you can center on a particular selection, which I will be covering in just a minute, or at any point you can reset it to how it was originally opened in order to get back to that. So using the Explore panel, we can easily hide or show different aspects of the structure. So for instance, if I wanted to see waters, I can easily turn them on, but we have them off by default since most people aren't interested in where the waters are. You can also hide different chains or select entire chains using this panel, this expand bar. So you can, for instance, hide chain B and its corresponding ligand with just 2 simple clicks. You can also select all of chain A then, and you center it around and click on the center on selection in order to make it your focal point of your rotation. In order to deselect it, all you need to do is double click in the structure view, and it will remove all selection for you. So today I want to show you how I would create an image using chain A's as an example. So the first thing that I would want to do is show off the secondary structure of this chain. So I can easily do that using our Features panel and expanding the KSD secondary structure, which provides me with the beta strands as well as the alpha helices. Using this, I can select all of the beta strands for both chain A and chain B, and then using the Style panel and the Color Expand bar, I can color these beta strands with a solid color, whereas by default it is on color by element, with gray being the color for carbon. So I want to fill it with a solid color. Easily do that and pick Orchid. And then in order to see the color change, I need to deselect it, so I double click in the structure view again. So I also want to color the alpha helices. So I can do that by selecting the helix from the feature panel, and again, simply fill it by a solid color, and I choose maroon. I also want to point out that throughout this entire presentation, every selection that I make, the Details panel will update in order to show the particular selection that I've made. So for instance, I have selected all of the alpha helices for chain A and chain B, which is designated here in the Details panel. If I clear the selection then, I then can see all of my alpha helices now in this maroon color and the beta sheet strands in the purple color. So for a presentation or a publication, I would most likely want to have a white background on my image instead of a black one. So I can easily change the background color by clicking this color well here, and choosing then white from the crayons. However, often when you change your background to white, you need to adjust your particular lighting. So we can easily do that using the lighting expand bar and expand that. And we have several options. And the first thing that tends to be an issue with white coloring is the depth cues, which is the fogginess of it. So you can easily just turn those off. If you still would like to adjust the lighting on the structure, you can add or subtract multiple lights, including you can have up to 3 different spotlights, and you can change the orientation in which the light is coming from using these gold spheres, and you can drag them around to pick the point at which the light you should originate from. You can also turn off any ambient light or change the brightness of the spotlights using the slider here, or the intensity of the ambient light by using this slider. You can also change the properties of the structure itself and how shiny it is, as well as how much the surface reflects. If you're not happy with any of the changes that you've made and you want to go back to how it originally was, you can easily do that by reverting back to the default settings by clicking that. So I'd like to learn a little bit more about the structure that I'm somewhat unfamiliar with. And the first thing that would come to mind is what is involved in the active site. So since I'm unfamiliar with this particular structure, I'm not quite sure which residues would be involved, but Protean 3D allows you to easily identify them using either the features panel, here it listed off our sites, or also in the sequence view, you can also see that the active site for chain A is listed by this label, as well as represented by these red cylinders. So you can easily select any of the red cylinders, which will select the entire active site. And I am interested in seeing these particular residues as balls, or as the entire side chains and not just the backbone ribbon. So I can easily change that using the rendering expand bar in the style panel and select side chains, which will change the rendering for my current selection. So I can change those to ball and stick. I'd also like to be able to designate these separate from the other coloration, so I'd like to color them to a light blue in order to identify them at a later time. And deselect to be able to see them. So if I zoom in here, you can see that all of the side chains now are pointed towards this ligand that appears to be bound in the active site. However, I want to be able to understand what the chemistry of these particular side chains are, which I can't do with my current coloration. So again, if I select the active site residues from the sequence view, I can change the color style to color by element except for carbon, which won't change my fill color, to be able to see the chemistry of each of them. So I can see now the nitrogens are blue and the oxygens are red, giving me a better indication of what their chemistry are. I also can easily select things from the structure view by double-clicking on anything. So for instance, if I want to double-click select my ligand, I can just double-click on that. Notice the details panel has now updated to give me more information about my particular ligand that I've selected. It provides me with a 2D structure, as well as biophysical properties about the ligand, as well as the B factor for how the ligand is bound and is ordered. The details panel also provides lots of information for other aspects. For instance, if you select an entire chain from the Explorer panel, you can find out the biophysical properties for an entire chain. So this is very helpful if you know that your protein, for purifying your protein, this would provide you easily with your molecular weight, as well as your extinction coefficient and what the protein would actually absorb at 280. If at any point you aren't familiar with any of these terms, you can easily select the question mark here, which would bring you to our help, and that will provide you with definitions for each of the values listed there. You can also select smaller regions, for instance, one particular helix, and you can also select individual residues to learn more about a particular residue's characteristic. For instance, if I was interested in this histidine in the active site, I could double-click on it and learn a little bit more about it. I could also, using a command click, select the ligand as well and determine how far apart the two are using the structural properties. And this tells me that the oxygen 1 from PGH, the ligand, as well as the epsilon 2 nitrogen from the histidine are 2.66 angstroms away from each other. So now that I've learned a little bit more about the structure and I want to maintain my colors, I can easily save this file as a .structure file using the File menu and Save As, and save it on my desktop as a .structure file that you can easily then open again in Protean 3D, and it will retain the orientation and the coloration that you have already designated. So I can find a orientation that I find the most appealing to present the information that I want, and then using the File and Export Image, I can export an image of a PNG or a JPEG that I can easily import into some sort of PowerPoint or presentation software that allows me to then present what I have discovered in Protean 3D.
Amanda, I'd like to take a minute or two for questions here, if this is a good time. One, you talked about the secondary structure. It looks like we have KSD secondary structure shown in the sequence view. Is that coming from the PDB file?
Actually, the KSD is a calculated secondary structure that we calculate when we open your file. The PDB secondary structure is what is designated in the PDB file. So even if your PDB file doesn't have a secondary structure, we will calculate one that is very similar to what is would be represented in your PDB file.
Great. And then another question about the rendering. You showed changing rendering on a selection. Is it possible to change the rendering on your entire structure?
Absolutely. It's not that difficult at all. In fact, Protean 3D, when you have absolutely nothing selected, equals everything is selected. So, for instance, I have nothing selected right now, and I can just go to ball and stick on all of my residues for the entire structure. You can also change the backbone from ribbon to tube or spheres or wireframe, or however you feel like presenting your structure.
Great, thank you.
So now that I've shown you a little bit about the coloration and renderings, I want to show you how to make surfaces. For that, I'd like to bring back chain B, and then again, with nothing selected, if I center on selection, it brings me back to the whole molecule being centered. So to create a surface, I simply expand the surface's expand bar, and I can just simply click the plus sign, or I can click on the arrow next to it, which provides me with the list of the different surfaces that we can create. We can create a molecular surface or a solvent-accessible surface with 3 different qualities for each. So for now, I'm going to create a molecular surface of normal quality, which is our standard, by just clicking on the plus sign. And since I have nothing selected, it creates a surface around everything. As you can see, what I mentioned before, the 2 different colorations, we have all the loops that are still in the default color, which is gray is carbon, and everything in the amino acid chemistry still being colored. Whereas this pink now is the fill color of the helices that I filled to be a solid color and not indicate the amino acid chemistry. So I can easily change that by changing everything to color by element except for carbon. So now that I have a surface, I would like to show about how the ligands bind into the active site. For instance, this particular ligand, I'd like to see what it would look like as a ball and stick. So I can change the ligand rendering from spheres to ball and stick and see that it fits very nicely into this pocket, but it appears to have quite a bit of space left over. Whereas if I rotate the molecule around and look at the ligand that I didn't change to a ball and stick and that is still in the spheres, you can see that it fills up the space a lot more, indicating that actually the ligand is probably bound quite tightly into the active site. Being interested in also what the solvent would be able to access, I can create a surface around a solvent-accessible surface. But I'd like to point out that Protean 3D, you don't have to just create a surface around just one particular thing. You can create around any type of selection. So for this, I'd like to create it around just chain B. And then I'd expand this list and choose normal quality from solvent accessible. And that creates it around just chain B. I can also, so I don't get confused, change the name of my surfaces. So for instance, this first one was a molecular surface, and I can easily name it that. By double-clicking in the name box, name column, I can change the surface name. And this was a solvent. I can also easily change the details in a similar fashion by double-clicking. So I want to be able to differentiate between these two surfaces a little bit better by coloring them a bit differently. So in order to color them differently, I need to color the residues underneath. So for instance, I would first color the molecular surface using the color expand bar, and I'd like to color it nice steel gray, which also colors my second surface because the residues underneath are the same. So to change only the residue color for chain B and not the molecular surface, then I simply click on this lock here, which locks the color for the molecular surface. I can now select my solvent accessible surface and change the color to a nice blue, which changes only that surface color and not the other one. You can also change the renderings of your surfaces as well as the qualities. So for instance, if I decided I didn't want this to be a solvent accessible surface, I can change it to a molecular surface and it would recalculate. I can also change it to a fast quality, which recalculates it. I can also change it from a solid to a mesh to be able to see through it, and now I can see my molecular surface underneath and see where my ligand is located. So if I zoom in on the ligand here, you can see that the solvent accessible surface doesn't appear to touch the ligand, indicating that the solvent would not likely be able to access the ligand all that easily. If I at any point want to hide either of my surfaces, I can simply uncheck this box next to the surface, or I can uncheck or check the general box that hides or shows all of my surfaces. So I don't lose my work. Of course, I always want to save, and once it's in a dot structure format, it's as easy as clicking save.
Amanda, I think we have a few questions now, if this is a good time. One, you mentioned briefly the different quality of surfaces. What is the difference in those? Is the high quality a long computational time, or what would be the reasoning for using a different quality surface?
Well, none of our surfaces take all that particularly long to calculate. It's actually the number of vertices that are shown. So for instance, with the mesh, if you show here in fast, it's the least amount of vertices, so the mesh is fairly see-through and transparent. However, if I move to a high quality, you can see that the increases number of vertices, so it just increases the number of points that you see, which if you are looking for a solid surface, it might be a smoother look instead of a little bit choppier with the fast quality. So that's the only difference between them.
Great, thank you. And so far, the whole demonstration has been using a PDB structure file. But we had a question come in. Let's say you're working with a DNA file in Lasergene, perhaps SeqBuilder or another application, and then you translate that to protein. Could that translated sequence be opened in Protean 3D, and then what kind of analyses could you do in that case?
Absolutely. We work with sequence-only files as well, which I'll show a little bit later in my presentation. And you would not see the structural information, obviously, because there wouldn't be any for your particular protein. However, what you can do is you can see the analysis view, and you can work with all of our prediction methods in order to learn more about your protein and what it might look like and whatnot. But for now, I'm going to move on to showing you how to learn a little bit more about your structure by aligning it to another structure. So again, I'm going to hide my surfaces here, and so I want to add another structure to this particular file. So I can do that by clicking File and Add to Document. And I can add my second file and navigate to it. So the structure I'm going to would like to align to is 3TA6, which is a similar protein that has the same function. However, it comes from a bacteria, whereas the one I have been working on is from a yeast. But I skipped ahead here. When I add to my document, I simply click on Add and select my file and click Open, and that puts it into a list that to be added. So because we can do multiple different types of opening and adding to a document, we have the options on how you would like to view the structure view. And by default, we center on screen so that for aligning to similar structures versus opening a large split entry or a biological assembly. But since I skipped ahead, I can just cancel out of this area. So I have my second structure open now in a new document that we switched the name to Untitled so you don't accidentally save over any of your previous work. And you can see the second structure is centered in the obscurely in the structure view, but you can also see that it is now listed in the molecule section as well as in the features panel. You have all of the features from the second structure as well. Additionally, the details panel shows all the same information that it showed previously with only one structure open, but now with two structures it has it in a slightly different organization so that you can identify what information belongs to what structure. So because I would like to just be able to identify this structure a little bit differently, I can select the entire thing from the Explorer panel and go to my color and change it to a nice pink to identify it differently from our 7-TIM. So I'd like to understand a little bit more about what chain of 3TA6 might be more similar to chain A of 7-TIM. So to do an alignment for those, I can simply select chain A and then using my command button also select chain A from 3TA6. From the structure menu, I then select align structures and rigid body alignment for selected regions. And that will align both of the chain A's to each other in the most appropriate manner with the least distance between each of the different atoms involved in the alignment. So in order to determine how good my alignment is, I can deselect everything by double-clicking again, and then in the details panel, it reports that chain A for 7-TIM versus chain A of 3TA6 has an RMSD, or root mean squared deviation, of 1.7. So I'd like to see how the number compares to the chain A of 7-TIM being aligned to chain B of 3TA6, which I can easily do by, again, doing my command selection in the Explore panel and then selecting Align Structures for the rigid body alignment. So the root mean squared deviation that I just showed is actually the least amount of distance, the average distance between each of the atoms that are involved in the alignment. So that is what the alignment is doing when it is aligning these structures, is finding the smallest average distance between each of the atoms in the alignment. So again, I deselect, and I now can see that chain A of 7-TIM versus chain B of 3TA6 has a root mean squared deviation of 1.69, so slightly better than chain A. But I know that these structures have, these two proteins have similar functions, and so I want to know if there's any particular conserved portions. So I know a little bit about this particular beta barrel that is in the center of this protein, so I want to see how the conserved region between the two proteins differs. So I want to select the beta strands for chain A of 7-TIM and of chain B from 3TA6. So using my features expand bar and command selection again, I select only that particular region. We can also do an alignment on just that particular selected region. And since it is smaller, it goes quite quickly, and I can deselect and see that the root mean squared deviation for the conserved beta barrel between these two proteins is only 0.6, indicating that evolutionarily this particular region of the proteins is highly conserved. So now that I've shown you a bit about how to align and create surfaces on different, I also want to show you more what I showed you a little bit before about the analysis view and how we can use the analysis view to learn more about protein, whether or not you have a structure, or whether or not you have a structure. So first I'd like to start with this file. And you can see that both proteins are also in the analysis view. You have 7-TIM, and over here you have 3TA6B. For ease of use, we cluster all sequences that are identical into one particular column. So as you can see, chain A and chain B of 7-TIM have identical sequences, as well as chain A and chain B of 3TA6. For ease of use, I only want to focus on 7-TIM, so I can just uncheck 3TA6 from the Explore panel, and that removes it from the analysis view as well. So the analysis view shows many predictive methods that we have listed in our methods panel. Most of these methods were also previously found in Protean, if anyone is familiar with the application of Protean that we also create. However, we've added some updates and some improvements, as well as a couple of new methods to our methods panel now. So the method I'd like to show you today with relation to a structure is our B-cell epitopes neoclone method. So most immunogenicity methods are generalized for any sort of immune response. Some are designed for particular cells, such as the T cells, but this method, the neoclone method, is specific for B cells, which are responsible for making antibodies. We developed this method recently with the help of a biotech company, Neoclone, that is also in Madison, Wisconsin, who specializes in the production of monoclonal antibodies. If you're interested in how this method compares to our competitors, we have a white paper that's available on our website. And this is our first release of the B-cell epitopes. In the future, we intend on improving and tweaking and making sure that this is the best B-cell epitope predictor out there. So all we have to do is simply check a box, and the method runs, and it appears in the analysis view. No other work is required. So our method here has a summary of both chains together as well as each chain individually. And if you expand it one more, you can see the levels that go into the synopsis levels. So we have our highly antigenic regions as well as our slightly lower antigenic regions. So you can see with our highly antigenic regions that you can pick out locations that might be of interest that have also other regions with it. For instance, my eye gets immediately drawn to this particular region that has multiple highly antigenic regions as well as a consistent prediction of a antigenic region. So I can select that, and it will select for just chain A, or I can also select it for both chain A and chain B. To identify where it is located in the structure, I can easily color it a nice pink color to draw it out. I'd also very interested in this larger region that has a highly antigenic region, and I can select it as well for both chain A and chain B, and then color it an orange to see approximately where it is located. So I can flip back to the structure view and see where these two regions are located. If I clear the selection, then I can see the orange region as well. So the first region that I selected and colored a pink clearly is on the surface and involved in this helix, whereas this beta strand is potentially a little bit more buried. In order to confirm that, I can easily make a surface around olive 7-Tim that is a solvent accessible surface, and I can see that clearly that pink region is solvent accessible, whereas the orange region is not quite as accessible. This tells me that the pink region would potentially be an excellent antigenic region for creating an antibody for a native protein, whereas the beta strand region would be potentially more useful for a denatured protein and for using in other methods such as Western blots. But as I mentioned before, you can also learn quite a bit about your structure without even you can learn far more about your protein sequence without having a structure. For instance, if I didn't have a structure that I could map out these particular regions to, I could use the surface probability method and select that in order to identify different regions if I'm looking for an antigenic region that happens to be on the surface. But I can also open any particular sequence-only file, which I would like to do now. I have chosen a portion of a yeast polymerase, so it's only one particular subunit, and this file in particular I downloaded straight from NCBI. But as you can see here, we have lots of different file formats that we can open, including those protein files from that you can create using other DNASTAR applications such as .pro. Those also can easily be opened in Protean 3D. So the major difference between opening a structure file and a sequence-only file is you don't have the structure view. But you can still do everything else that you could do with the structure file, with the analysis view. So a couple of methods that might be particularly interesting for a protein that you aren't familiar with all that much or want to learn a little bit more about is predicting different domains and sites using our ProSite method. So all you have to do is check the box next to it and the method runs. This method is searching through a large library of different domains and sites that have been predicted based off of sequences. ProCite is available via Exposé, and you can learn more about the method using our help, which again will link you to more about this particular method. So ProCite now has predicted that this has a ZF-FAP domain, but I'm not quite sure what that is. So by selecting it in the analysis view, it selects the particular region of the domain, and then the details panel provides me with more information. So I see here from the summary that it is a zinc finger SAP type profile, which indicates to me that this might have a zinc finger type domain, which would make potentially sense for it being part of a yeast polymerase. You can also change to see all of the results that ProCite predicts, including the frequent ones, by selecting the method from the methods panel and then expanding the parameters panel. The parameters panel is dynamic and will change based off of which method you have selected. For instance, if I select charge density, it will show the parameters for that particular. So for ProCite, we by default hide the frequent patterns, but you can show those as pins here, and they will include different sites such as potential glycosylation sites as well as phosphorylation sites. And you can, again, learn more information about each of those by selecting the pin from the analysis view, and again, the details panel then shows you more information. Also, you can click the action out of the details panel to see the documentation that ProCite provides on that particular domain or site. Another particularly interesting method that is new to 10.1 is our proteases method, which helps you predict any potential cleavage sites. So when you check the method, you can see some of our potential proteases. Now, if you are interested in those that don't cut, you can easily see those by exporting the full protease report to a file, and those will include all of the proteases and how many times they cut, including those that don't cut at all. You can also change the filter and see how different types of proteases by, again, selecting the method from the methods panel and then changing the parameters. And we've included different filters that including chemicals or those that cut infrequently, which is our default, which shows that cut at least once but no more than 5. You can also change that using a custom filter to change it to cut no more than, for instance, 10 times. And then the analysis view immediately updates with proteases that cut at least once but no more than 10 times. If you want to find more information about any of these particular proteases, you can select them from the analysis view. And again, the details panel will provide more information about the particular site that it cuts, as well as PubChem number or MEROPS number, depending on if it's an enzyme or a chemical. So that brings me to the end of the analysis view section. If you have any questions about that, I would be happy to answer them.
Yes, we do have a couple questions. You may have mentioned this, but when you were talking about the neoclone B-cell epitope prediction method, do you have any ideas or data on how that compares to other available methods?
We do have a white paper that is available on our website, the DNASTAR website, on the Protean 3D page that provides information about how we compare to, so this PDF right here, the white paper, compares our B-cell epitope prediction to our competitors, if you would like to read that at any time.
Great. And the other question is there any editing capability for the sequence data in Protean 3D or elsewhere in Lasergene?
You can easily edit your sequence, for instance, using SeqBuilder or one of our other applications, but at this time, Protean 3D only maintains the sequence of the file that you load in. It doesn't allow you to edit them at all. but you can do that with other applications such as Seq Builder or Megaline.
Okay, great. Thank you.
So for my final section, I wanted to show how you can view a molecular morph, which is by opening, first you can open from our motion library. We have acquired these particular morphs from the Yale Morph server. However, you can also create your own molecular morph by submitting structures that you're interested in to the Yale Morph server, and then download that file and open it here. What they do at the Yale Morph server is they create chemically plausible transitions between same proteins of different conformations. So here in our motion library, we have 393 different unique motions, and that provide lots of information about what the protein might be doing. To be able to easily find the protein that you're interested in, you can simply search using our search box here. And for instance, today I'm going to search for something that binds a UDP moiety. So this gives me 6 different unique motions, and today I'm going to look at 1EE to 2UAG. And I just double click on it. And if it's not previously downloaded, it will prompt you to download. But I had previously downloaded this file, so it opens right away. So again, I can just rotate the structure around in order to find a orientation that allows me to learn the most about this protein. So it appears that there are 2 different domains, a top domain and a larger domain on the bottom, that appear to be moving along a hinge. So if I want to learn a little bit more about that, I can stop any of the motion at any time or see any of my different models using the models panel. And so I want to select let's select the bottom region on the N-terminal domain from the sequence view, and I can determine approximately the whole domain. So I like to then color it a nice blue to identify it. And then I'm interested in this top domain as well, which appears to start at this next beta strand, which I can identify easily in the sequence view as right here, and then select it to the end of this C-terminus as well. And to identify it, I'll color it a nice green. So now by starting my motion again, can see my 2 different domains easily moving about each other. But I would also like to be able to present that where the hinge is located, which I can identify clearly between the 2 color regions, which which are marked in the sequence view as well, and color it a nice pink color. So now that I can present easily where my hinge is and my 2 different domains, I'd also like to see if there's anything else that I might be interested in demonstrating in my presentation. So, for instance, there appears to be a loop here that is interacting with this helix. So I'd like to identify that particular loop by double-clicking and selecting it in the structure view to give me an idea on where it might be located in the sequence view, since the selection is universal. So I see it here, and then I can select the entire loop and color it, for instance, orange. But I'm also interested in the helix that it's interacting with. So if I double click again in the structure view and select the portion of the helix, it brings me to the helix that it would be involved in here in the sequence view. And since my secondary structure is here, I can easily select that to the portion of the helix that I'm interested in. And again, color it a nice orange. Using my models panel then, I can start my motion up, and now I can easily see that the two different domains, as well as any portions that happen to be moving directly that I would like to present. In order to use this movie in some sort of presentation software, I can use File and Export Movie, and this will export it to a .mov or a .avi file that is easily usable in any sort of presentation software. With that, that concludes my portion. If you have any questions, I'd be more than welcome to answer them.
Thanks, Amanda. We will let people have a few minutes to chat in any last-minute questions. While we're waiting, I'll go ahead and take care of a few administrative items. A couple of you asked me about the webinar recording. That will be available on our website and on our YouTube channel, usually within a day of the presentation. We just need to get it downloaded from the WebEx site and then uploaded to our site. So all of you will receive an email with the details on how to access that. And beyond that, we also have a large collection of videos on our website. So if you want to learn more about Protean 3D or any of our other applications, we've got over 100 videos in our video library, and that URL should be up on your screen now. That's DNASTAR.com/videos. And that's got usually 3 to 5 minute videos on a specific workflow, including some of the things Amanda covered today. So epitope prediction, applying surfaces, using the analysis methods, and so forth. And then our next webinar is on December 5th at the same time. It's also a Wednesday, and the topic there is automated bacterial genome closure using our Lasergene genomics suite. That's going to be presented by our own Matt Kaiser. So if you want to learn more about some of our next-gen capabilities, we hope you can join us for that webinar as well.